In this, we are going to learn about what is recombinant DNA. So, recombinant DNA is also known as Invitro recombination. A cloning vector is a DNA molecule that carries foreign DNA into host cell. The other way, cloning vector is a DNA molecule which carries foreign DNA molecule into host cell, where it replicates, producing many copies of itself. Host cell replicate ಆದಾಗ ಅದು it produces many copies of itself along with the foreign DNA. There are many types of cloning vectors such as plasmid and phages. So ಇದನ್ನು ಓದ್ತಿರೋದೇನೆಂದರೆ ನಿಮಗೆ concentrated two vectors, plasmids, enzymes used in R-DNA technology, steps in R-DNA technology, isolation and extraction of foreign DNA, extraction of plasmid DNA, restriction, digestion and ligation of foreign DNA with vector DNA, production of R-DNA. So what are vectors? Basically vectors are small, extra chromosomal, self-replicating and circular DNA molecules. So ಪ್ರತಿಯೊಂದಕ್ಕೂ ಅದರದೇ ಆದಂಥ ಇಂಪಾರ್ಟೆಂಟ್ ಫೀಚರ್ಸ್ ಇದೆ. ಸೊ ವೆಕ್ಟರ್ಸ್ ಇಂಪಾರ್ಟೆಂಟ್ ಫೀಚರ್ಸ್ ಬಂದ್ಬಿಟ್ಟು ದೇ ಆರ್ ಸ್ಮಾಲ್ ಆ್ಯಂಡ್ ಈಸಿ ಟು ಐಸೋಲೇಟ್. ಅದು ತುಂಬ ಚಿಕ್ಕದಾಗಿರುತ್ತೆ ಆ್ಯಂಡ್ ಐಸೋಲೇಟ್ ಮಾಡೋಕ್ಕೆ ತುಂಬ ಈಸಿಯಾಗಿರುತ್ತೆ. ಕೇಪಬಲ್ ಆಫ್ ರೆಪ್ಲಿಕೇಟಿಂಗ್ ಇನ್ ಹೋಸ್ಟ್ ಸೆಲ್ ರೆಪ್ಲಿಕೇಟ್ ಮಾಡೋಕ್ಕೆ ತುಂಬ ಕೇಪಬಲ್ ಆಗಿರುತ್ತೆ. ಇಟ್ ಹ್ಯಾಸ್ ಯೂನಿಕ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಕ್ಲೀವೇಜ್ ಸೈಟ್ ತುಂಬ ಯೂನಿಕ್ ಆಗಿರುತ್ತೆ. Contains several marker genes which facilitates screening of transformed cells. Contain control elements. Control elements ಏನಂದ್ರೆ promoters and terminators for expression of clone genes. Clone genes expression ಗೋಸ್ಕರ ಈ promoters, terminators and so on use ಮಾಡ್ತಾರೆ. So different types of DNA. Bacterial, artificial, artificial DNA, chromosomes and each artificial chromosomes, expressions, plasmids, bacteriophages and plasmids. So what are plasmids? They are self-replicating, small, double-stranded, circular DNA molecules. They are mostly present in bacteria and yeast or even in mitochondrial of eukaryotic cells. They carry genes for resistance in antibiotics, toxins and toxic materials. Their usually size is 1 to 200 kb. For example, you can use, it's mostly common in PBr 322 and PUC 19. And so PBr 322 refers, like P refers to plasmid, and BR refers to the scientist who discovered it. Is B refers to Bolivar, and R refers to Rodilger, and 322 where it refers to the it is unique from other plasmids. Same in the case of PUC 19. So what are bacteriophages?
Bacteria which infects virus.
Okay. So yeah, exactly. There are which viruses that infects bacteria. The most common are bacteria phage lambda and the phage M13 are used as cloning vehicles. So you can see over here, ಮೇಲ್ಗಡೆ phage lambda ಇದೆ. So ಕೆಳಗಡೆ diagram ಇರೋದು M13 structure. So some terms in R-DNA technology. So in that foreign or passenger DNA is a fragment of DNA molecule which is enzymatically isolated and cloned from a particular species. Foreign DNA on fragment DNA molecule ಆಗಿರುತ್ತೆ, in which enzymatically isolated and cloned, clone ಮಾಡಿ from a particular species ಆಗಿರುತ್ತೆ. Recombinant DNA or R-DNA is general name for a piece of DNA that has been created by combinations of at least two strands in the R-DNA form ಆಗಿರುತ್ತೆ. So here we can see the recombinant DNA technology. So recombinant DNA technology refers to joining of together of DNA molecules from two different species. ಎರಡು ಡಿಫ್ರೆಂಟ್ ಸ್ಪೀಶೀಸ್ನ ಜಾಯ್ನಿಂಗ್ ಮಾಡೋದು ಜೊತೆಗೆ ದಟ್ ಆರ್ ಇನ್ಸರ್ಟೆಡ್ ಇಂಟು ಹೋಸ್ಟ್ ಆರ್ಗನಿಸಮ್. ಆ ಜಾಯ್ನ್ ಆಗಿರುತ್ತಲ್ಲ ಅದನ್ನು ಹೋಸ್ಟ್ ಆರ್ಗನಿಸಮ್ಸ್ಗೆ ಇನ್ಸರ್ಟ್ ಮಾಡೋದು ಟು ಪ್ರೊಡ್ಯೂಸ್ ನ್ಯೂ ಜೆನೆಟಿಕ್ ಕಾಂಬಿನೇಷನ್ಸ್. ಹೊಸ ಜೆನೆಟಿಕ್ ಕಾಂಬಿನೇಷನ್ಸ್ನ ಮೇನ್ ನಾವಿಲ್ಲಿ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡ್ತಿರೋದು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋಕ್ಕೆ. ಸೊ ಜೆನೆಟಿಕ್ ಕಾಂಬಿನೇಷನ್ಸ್ ದಟ್ ವ್ಯಾಲ್ಯೂ ಟು ಸೈನ್ಸ್, ಮೆಡಿಸಿನ್, ಅಗ್ರಿಕಲ್ಚರ್, ಇಂಡಸ್ಟ್ರಿ, ಸೊ ಇವೆಲ್ಲ ಫೀಲ್ಡ್ಸ್ಗೆ ಯೂಸ್ಫುಲ್ ಆಗಿರೋ ಥರ ಹೊಸ ಒಂದು ಪ್ರಾಡಕ್ಟ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬೇಕು. ಸೊ ಇದು ಪ್ರೊಸೀಜರ್ ವೇರ್ ವಿ ಕ್ಯಾನ್ ಸಿ ದ process of recombinant DNA technology. Okay. So enzymes in recombinant DNA technology. ಇದರಲ್ಲಿ ಬೇಸಿಕಲಿ ಮೇನ್ಲಿ ಮೂರು ಬರುತ್ತೆ. ಒಂದು ನ್ಯೂಕ್ಲಿಯಸಸ್, DNA modifiers and DNA ligases. ನ್ಯೂಕ್ಲಿಯಸಸ್ ಅಲ್ಲಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಫಸ್ಟ್ ಒನ್, ಸೆಕೆಂಡ್ ಒನ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸ್ and third one ribonucleus. DNA modifiers ಅಲ್ಲಿ DNA polymerase, reverse transcriptase, alkaline phosphates, polynucleotide kinase, terminal nucleotidal transferase, methyl transferase. So third one DNA ligases ಅಲ್ಲಿ DNA ligase. So steps in DNA R-DNA technology. So there are basically 4 steps. The step one is isolation of foreign DNA from a particular species. One particular species in the foreign DNA na isolate ಮಾಡಬೇಕು. Then restriction enzyme digestion, ligation of DNA molecules and insertion of R-DNA into host. R-DNA ಇರುತ್ತೆ ಅದನ್ನು host ಗೆ insert ಮಾಡಬೇಕು. Isolation and preparation of foreign DNA. To extract DNA from a bacterial cell, the cells are treated with enzyme called lysozyme. DNA ನ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡೋಕ್ಕೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಇಂದ ಸೆಲ್ಸ್ ನ ಎಂಜೈಮ್ ಕಾಲ್ಡ್ ಲೈಸೋಸೈಮ್ ಇಂದ ಟ್ರೀಟ್ ಮಾಡಬೇಕು ಇನ್ ಆರ್ಡರ್ ಟು ವೀಕನ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ವಾಲ್. ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ವಾಲ್ ಇರುತ್ತೆ, ಸೊ ಅದನ್ನು ವೀಕನ್ ಮಾಡೋಕ್ಕೆ ಲೈಸೋಸೈಮ್ ನ ಯೂಸ್ ಮಾಡಬೇಕು. The weakened bacterial cell wall is subsequently treated with an anionic detergent such as sodium didoxyl sulfate (SDS). Bacterial cell wall ಏನಿರುತ್ತಲ್ಲ ಅದು subsequently treat ಮಾಡೋದು with an anionic detergent. So it is called as sodium didoxyl sulfate (SDS). Eukaryotic plant cell possess a rigid cell wall. Usually, eukaryotic plant cell rigid cell wall possess ಮಾಡುತ್ತೆ. So that the cells are treated with the enzyme cellulase to rupture the cell wall. Cell wall na rupture ಮಾಡಕ್ಕೆ cellulase ಅನ್ನೋ enzyme ನ ಯೂಸ್ ಮಾಡ್ತೀವಿ. Eukaryotic animal cells do not possess cell wall. Cell wall possess ಮಾಡಲ್ಲ, but cell membranes to isolate the DNA. The cells are treated with anionic detergents such as SDS solubilize the cell membrane. Steps in R-DNA technology. Steps main ನಾವು ಅವಾಗಲೇ ಏನು ಸ್ಟೆಪ್ಸ್ ಓದಿದ್ರು ಅದು ಇವಾಗ ಬ್ರೀಫ್ ಡೀಟೇಲ್ ಆಗಿ. ಸೊ ಐಸೋಲೇಷನ್ ಆಫ್ ಫಾರಿನ್ DNA ಫ್ರಮ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಪೀಶೀಸ್. ಅದರಲ್ಲಿ ಫಸ್ಟ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಷನ್ ಆಫ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ DNA. Bacterial cells are added into extraction medium. ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಸ್ ಇರುತ್ತಲ್ಲ ಅದನ್ನ ನಾವು ಎಕ್ಸ್ಟ್ರಾಕ್ಷನ್ ಮೀಡಿಯಂ ಆಡ್ ಮಾಡ್ಕೋಬೇಕು. ಕಂಟೈನಿಂಗ್ ಸೆಲೈನ್ EDTA ಲೈಸೋಸೈಮ್ ಇಸ್ ಆಡೆಡ್. ಸೆಲೈನ್ EDTA ಲೈಸೋಸೈಮ್ ಅದರಲ್ಲಿ ಕಂಟೈನ್ ಆಗಿರಬೇಕು. and 37 degrees for 20 minutes in water bath. Then addition of 10% of SDS after incubation of 60 degree Celsius in 10 minutes. It is then cooled on ice. ಇನ್ಕ್ಯುಬೇಷನ್ ಆದ್ಮೇಲೆ 60 ಡಿಗ್ರೀಸ್ 10 ಮಿನಿಟ್ಸ್ಗೆ ಅದನ್ನ ಆಮೇಲೆ ನಾವು ಐಸಲ್ಲಿ ಇಟ್ಟು ಕೂಲ್ ಮಾಡಬೇಕಾಗುತ್ತೆ. ಫಸ್ಟ್ ಎಥನಾಲ್ ಪ್ರೆಸಿಪಿಟೇಷನ್. ಇದರಲ್ಲಿ ನಾವು ಫಸ್ಟು ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡ್ಕೋಬೇಕಾಗುತ್ತೆ. ಎಷ್ಟಕ್ಕೆ 10,000 RPM. ರಿಮೂವಲ್ ಆಫ್ ಅಪ್ಪರ್ ಅಕ್ವೇಸ್ ಲೇಯರ್ and it keeps and is kept in a beaker and the solution is overlaid with 80% of ethanol. Removal of upper aqueous layer. Upper aqueous layer na remove ಮಾಡಕ್ಕೆ and it is kept in a beaker and in a solution of overlaid of 80% ethanol. So ಇಲ್ಲಿ 80% ethanol ನ ಯೂಸ್ ಮಾಡ್ತೀವಿ. Two layers are straightened with the help of a glass rod and the DNA fibers is wound out on the rod. So two layers ನ ಸ್ಟ್ರೇಟ್ ಮಾಡಿದಾಗ ಗ್ಲಾಸ್ ರೋಡ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಅದು ಏನಾಗುತ್ತೆ DNA ಫೈಬರ್ಸ್ ಗ್ಲಾಸ್ ರೋಡ್ಗೆ ಸುತ್ತಕೊಂಡಿರುತ್ತೆ. ದೇ wound on the glass rod. Remove the solvent. Finally the fibers DNA is blotted using filter paper. Solvent ನ ರಿಮೂವ್ ಮಾಡ್ಬಿಟ್ಟು ಫೈನಲ್ DNA ಫೈಬರ್ಸ್ DNA ಏನಿರುತ್ತಲ್ಲ ಅದನ್ನು ಬ್ಲಾಟ್ ಮಾಡಬೇಕು ಯೂಸಿಂಗ್ ಫಿಲ್ಟರ್ ಪೇಪರ್. RNA treatment and second ethanol precipitation. Addition of saline sodium solution consisting of sodium acetate. It should be done in 37 degrees Celsius for 30 minutes. Okay. Incubation of RNAs with addition of first one sodium chloride solution, second one isomyl alcohol. Solvent removed by 80% ethanol and DNA is spooled out by glass rod, purified and stored in 40 degrees Celsius. So solvent removed by 80% of ethanol. 80% of ethanol use ಮಾಡಿಕೊಂಡು DNA is pulled out from glass rod and ಅದನ್ನ -40 degrees Celsius ಅಲ್ಲಿ ಸ್ಟೋರ್ ಮಾಡಿರೋದು. Isopropanol precipitation. So addition of isopropyl alcohol. DNA is redissolved in sodium saline citrate and stored in -40 degrees Celsius. So DNA is redissolved in sodium saline citrate. DNA ನ redissolve ಮಾಡಬೇಕು. redissolve ಮಾಡಿಬಿಟ್ಟು ಅದನ್ನ -40 degrees Celsius ಅಲ್ಲಿ ಸ್ಟೋರ್ ಮಾಡಬೇಕು. Then assay for nucleic acid. So other spectral analysis in that 1 kg per ml of DNA gives absorbance of 2.0 at 260 nanometer in 1 centimeter weight. The ratio of epsilon 260 into epsilon 280 should be between 1.8 to 1.9 of pure DNA. ಒಂದು ರೇಷಿಯೋ ಇರಬೇಕು. ರೇಷಿಯೋ and the value less than 1.75 indicates protein contamination. Value 1.7 1.75 ಇನ್ನ ಕಮ್ಮಿ ಇಂಡಿಕೇಟ್ ಮಾಡಿದ್ರೆ ಸೊ ಪ್ರೋಟೀನ್ ಕಂಟಾಮಿನೇಷನ್ ಅಂತ ಹೇಳ್ಬೋದು. Extraction of plasmid DNA. Culture of cells in liquid medium. The cells are harvested in a cell extract is prepared. The extract of deproteinized the RNA is removed from DNA is concentrated by ethanol precipitation. The extract of deproteinized extract idea. The DNA is removed and the RNA is removed and the DNA is concentrated by ethanol precipitation. DNA na concentrate ಮಾಡಬೇಕು ethanol precipitation ಅಲ್ಲಿ. So separation of the plasmid DNA from the large bacterial chromosomal DNA. So separation of the basis of size. Cell lysis. The DNA fragments are removed with cell debris by centrifugation. So cell debris na ಹೇಗೆ remove ಮಾಡ್ತಾರೆ by centrifugation. Cells are treated with EDTA and lysozyme. After centrifugation, a cleared lysate consists almost entirely of plasmid DNA. Centrifugation ಆದ್ಮೇಲೆ cleared lysate almost entire plasmid DNA consist ಮಾಡುತ್ತೆ. So separation on the basis of conformation by alkaline denaturation. So addition of sodium hydroxide of pH 12 to 12.5. If sodium hydroxide pH level 12 to 12.5 add ಮಾಡಬೇಕು. So if acid is added, the denaturated bacterial DNA plasmid will reaggregate in mass. So reform ಆಗುತ್ತೆ. So insoluble network can be chelated by centrifugation, leaving plasmid DNA in the supernatant RNA, and protein can be removed by centrifugation. Protein na centrifugation ಇಂದ remove ಮಾಡಬಹುದು. digestion of DNA with type 2 of restriction enzyme staggered on blunt ends or created. Most commonly, both donor DNA and vector DNA are digested with the use of same restriction enzyme that produces sticky ends of vector and donor DNA to bind to each other and form recombinant molecules. So basically, most commonly the donor DNA and the vector DNA digest with the use of same restriction enzyme. Using the same restriction enzyme, they digest and produce sticky ends of vector and donor DNA to bind to each other and form recombinant molecules. A plasmid vector carries a single E. coli restriction site. So digestion with the restriction enzyme E. coli converts the circular DNA into a linear molecule with sticky ends. So plasmid vector usually carries a single E. coli restriction. So it basically digests with the restriction enzyme E. coli, and it further converts into circular DNA into linear molecules. Donor DNA from any other source also treated with E. coli enzymes to produce donor DNA. Donor DNA from any other source are treated. Where other source treat ಮಾಡ್ತಾರೆ with the E. coli enzyme to produce a population of fragment of a donor carrying the same sticky end. The same sticky end ಇರಂತದನ್ನ produce ಮಾಡಬೇಕು. When the two population of mixed DNA fragments from the source can be unite because double helix from between their sticky ends. There are many opened up vectors molecules in the solution and make and many different E. coli fragments of donor DNA. Therefore, the diverse array of vectors carrying a different donor inserted will be produced. They will be produced in a different way. At this stage, although sticky ends have united to generate a population of chimeric molecules, the sugar phosphate backbones are still not complete at two positions at each junctions. Each junctions ಅಲ್ಲಿ ಅದನ್ನ ಕಂಪ್ಲೀಟ್ ಆಗಿರಲ್ಲ. However, the backbones can be sealed by addition of enzyme DNA ligase. So backbone na seal ಮಾಡಿರಬಹುದು addition of DNA ligases ಅಲ್ಲಿ, which creates phosphodiester bonds at junction. Certain ligases are even capable of joining DNA fragments with blunt contents. Next we are coming to production of R-DNA. ಹೇಗೆ R-DNA production ಆಗುತ್ತೆ? So select donor DNA and vector DNA. First ನಾವು donor DNA and vector DNA ನಾವು select ಮಾಡ್ಕೋಬೇಕು and then purify a gene sequence to be transferred. Gene sequence ಏನು ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕಲ್ಲ ಅದನ್ನ ನೆಕ್ಸ್ಟ್ ನಾವು ಪ್ಯೂರಿಫೈ ಮಾಡ್ಕೋಬೇಕು. Cut the larger donor DNA and vector DNA with the same restriction enzyme. Same restriction enzyme ಅಲ್ಲಿ larger donor DNA ಮತ್ತು vector DNA ಕಟ್ ಮಾಡ್ಕೋಬೇಕು. Modification of patents, mix donor DNA and vector DNA and add T4 DNA ligase. Mix donor DNA ಮತ್ತು vector DNA mix ಮಾಡಿದ್ಮೇಲೆ we should add T4 DNA ligase. Next, amplify R-DNA E. coli cells. The screened clone are transferred into fresh culture plates and let it grow in large population. So screened clones ಆಗಿರುತ್ತಲ್ಲ ಅದನ್ನ ಟ್ರಾನ್ಸ್ಫರ್ ನೆಕ್ಸ್ಟ್ ಫರ್ದರ್ ಕ್ಲೀನ್ ಆಗಿರೋ ಒಂದು ಕಲ್ಚರ್ ಪ್ಲೇಟ್ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕು. ಯಾಕೆಂದರೆ ಬಿಕಾಸ್ ಗ್ರೋ ಆಗುತ್ತೆ ಲಾರ್ಜ್ ಪಾಪ್ಯುಲೇಷನ್ಗೆ. Screen clones containing R-DNA. Extraction of R-DNA from cells are purified. Use the R-DNA for various generic appliances. So, thank you.
ಥ್ಯಾಂಕ್ ಯು. ಸ್ಲೈಡಲ್ಲಿದೆ. ಯಾರು ಹೇಳಿ ಕ್ಲಾಸ್ ತಗೋಕ್ಕೆ? ಕ್ಲಾಸ್ ತರೋಕ್ಕೆ ಕ್ವಶನ್ ಬರಬೇಕು ಇವಾಗ. ಥ್ಯಾಂಕ್ ಯು ಸ್ಲೈಡಲ್ಲಿದೆ. ಥ್ಯಾಂಕ್ ಯು ಹಾಕಿಲ್ವಾ? ಕ್ಲೋಸ್ ಮಾಡ್ಕೊ.